Hi viewers, today we are going to learn the antigen and antibody reactions. Under this topic, we are going to discuss the introduction to the topic and the related terms of the antigen antibody reactions like affinity, avidity, and cross reactivity, antigen antibody reactions in vivo, and antigen antibody reactions in vitro like agglutination, precipitation, neutralization, complement fixation, and Elisa. Let us start with the introduction to the topic. Antibody is an immunoglobulin molecule produced by the B lymphocytes of an organism when it is exposed to a foreign substance which is known as antigen. The interaction between these two molecules is called as the antigen antibody reactions. These reactions form the basis for humoral immunity, or we may also call it as antibody mediated immune response in vivo conditions, that is within the animal body. The study of antigen antibody reactions in vitro conditions, that is outside the body of the animal, otherwise in the laboratory, that is known as serology. It is very important in diagnosis of various infectious diseases, and identification and classification of antigens is the serotyping of various microorganisms by the use of specific anti-sera. The antigen antibody interaction is a bimolecular interaction, just like enzyme and substrate, but it is reversible in nature. This interaction involves non-covalent interaction such as hydrogen bonding, hydrophobic interaction, ionic bonding, and van der Waals forces. The interaction of antigen with antibodies may result in a variety of consequences, including agglutination if the antigen is particulate, precipitation if the antigen is soluble, activation of complement, neutralization, and other things. There are no such type of reactions when antibody with more antigen binding site reacted with heptane. There are no interactions when antibody and antigen are univalent. Cross-linking is possible if the antigen is multivalent and the antibody is divalent. No cross-linking is possible if the antigen of the antibody is univalent. And if you observe the primary interactions between the antigen and antibody, no covalent bonds are involved in the interaction between antigen and antibody, that is antibody and an epitope. The binding forces are relatively weak. They consist many of van der Waals forces, electrostatic forces, and hydrostatic forces, all of which require a very close proximity between the interacting moieties. Thus, the interaction requires a very close fit between an epitope and the antibody, like a lock and key fit, like that we will see in the enzymes. Because of the low levels of energy involved in the interactions between antigen and antibody, therefore, antigen and antibody complexes are readily to dissociated. And let us see some sort of features of the antigen antibody reactions. The reaction is specific. Entire molecules react and not the fragments. There is no denaturation of the antigen or antibody during this reaction. The combination occurs at the surface, so surface antigens are immunologically relevant. The combination is firm but reversible. The firmness is influenced by the affinity and avidity of the reaction. Antigens and antibodies can combine in varying proportions. Both antigens and antibodies are multivalent. Let us see the definitions of affinity and avidity. Affinity is the strength of non-covalent interactions between a single epitope and its paratope. Epitope is a part of antigen, whereas a paratope is the complementary part which binds to the epitope. It is a part of the antibody. So it is known as the affinity of antibody. The strength of non-covalent interaction between a single epitope and its paratope is the affinity of an antibody. Whereas the avidity, the cumulative strength of the multiple interactions between a multivalent antigen and antibody is the avidity of the antibody. So whenever you talk about the affinity, that is the interaction between a single epitope of the antigen and a paratope of the antibody, whereas the avidity, it is a cumulative strength of all the epitopes of an antigen and all the antibodies that are bound to epitopes of the antigen. Let us see some illustrations regarding affinity and avidity. See, here we are observing the high affinity. So this is the blue color one is the antigen and the orange color one is the antibody. Here there is more affinity. So antigen and antibody move towards each other. Whereas here you can see low affinity. So whenever you compare the high affinity, the antigen and antibody move towards each other, but here such a kind of phenomena is not at all seen. They do not move towards each other, they are going away from each other. So that is low affinity. And affinity of interaction. So the combined strength of non-covalent interaction between a single antigen binding site on an antibody and a single epitope is the affinity of an antibody. The association between a binding site and antibody with a non-covalent antigen is described as antigen plus antibody. Uses the formation of antigen and antibody, it is a reversible reaction. Whenever you see this as the affinity constant, it is demonstrated as the Ka, the ratio of forward to the reversible reaction. The affinity constant can be measured by carrying out equilibrium dialysis. Equilibrium dialysis. A dialysis chamber separated into two compartments by a semi-permeable membrane is taken in one compartment, antibody placed, and in another compartment radiolabeled ligand is placed. Affinity constant Ka is equal to Ag AB divided by Ag and AB. So here we can see the equation where Kj is the association constant to measure the affinity. See here, this is the antibody and this is the antigen. Here there is a reversible reaction. AgAb complex has been formed. See here, the equation is justified like this. Affinity constant is equal to AbAg, antigen antibody complex by individual AB and Ag, antibody and antigen. So K can be determined, constant can be determined by using equilibrium dialysis. Coming to the avidity. Avidity is the overall strength of binding between an antigen with many determinants and multivalent antibodies. See here it is showing affinity, one antigen epitope and one paratope of the antibody. Whereas the avidity, it is a combined effect of all the antigens and all the antibodies. If you take into consideration the antibody like IgM, immunoglobulin M, you can see these all are the epitopes and these are the paratopes of different antibodies of a multivalent antibody. So that shows the avidity of the antibody. See here, IgM, it is a pentameric form which has a high avidity with more paratopes which can bind many number of epitopes, whereas IgG, it is a monomeric form. Okay, it has a high affinity, that is tighter binding with the epitope of an antigen. And coming to the cross reactivity. Cross reactivity, it occurs when the two antigens share identical epitopes and the antibody will be cross reacted with it. For example, Treponema pallidum cardiolipin shares identical epitopes. The antibody cross reactive with both the epitopes, as well as other example is the cowpox and smallpox viruses shared identical epitopes. When a person is infected with a cowpox, becomes resistant to smallpox because of cross reactivity. Similarly, rickettsia is a obligate intracellular parasite share identical epitope with Protea vulgaris, and the rheumatic fever caused by the Streptococcus pyogenes is due to the cross reactivity of the M protein shares the identical epitopes with heart myocardium. This is in contrast, antibodies to A, B, O blood groups is due to cross reactivity. The intestinal microflora share identical epitopes of the A and B antigens. As the person having blood group A have anti-B, it is due to cross reactivity with intestinal microflora having the B antigen. Here we are observing the antibody, anti-X antibody, the antibody against the X structure. Here the same antibody can interact with the antigen Y due to the structural similarity. It is also can capable to interact with the antigen Z due to the similarity in the epitopes of the antibody. And coming to the antigen antibody interactions in vivo. So this is the main source, otherwise the central dogma for the humoral immunity. The interactions between the antigen and antibody are the central dogma of antibody mediated immunity, that is humoral immunity inside the body of the host. As a result of these interactions, the antigen is altered. That may result in the activation of antigen, the death of pathogenic microbes possessing the antigen, and there will be enhancement in the susceptibility of the antigen also. Then antigen antibody interactions inside the human body. So here neutralization, immune complex formation, then some of the times complement fixation, antibody dependent cell mediated cytotoxicity. ADCC means antibody dependent cell mediated cytotoxicity. And also some of the times we may observe hypersensitivity, autoimmunity, and the immunodeficiencies. And coming to the antigen antibody interactions in vitro conditions in the laboratory. Antigen antibody interactions in vitro, that is under the laboratory conditions, are referred to as the serological reactions or serology. Antigen antibody interactions were first adapted to laboratory tests to diagnose diseases in the late 1800s. At present, they have become a highly sophisticated and often automated discipline of immunology and are widely used in clinical diagnosis, epidemiology, and basic research. A variety of serological reactions are used in laboratories. The most important and often used ones are agglutination, precipitation, neutralization, complement fixation, and Elisa. Let us see one by one. Agglutination reactions. See here there is a clear-cut difference among all these five, whether that is agglutination, precipitation, and all these. Let us start with the agglutination reaction. The reaction of antibody with a multivalent antigen, that is always particulate antigen, insoluble antigen. Whenever the antibody interacts with the insoluble or particulate antigen results in the cross-linking of the various antigen particles by the antibodies. This cross-linking eventually results in the clumping or agglutination of the antigen particles by the antibodies. The antibodies play a major role in binding of the particulate antigens. For the first time, two bacteriologists, namely Herbert Edward Durham and Max von Gruber, they discovered a specific agglutination in the year 1896. The clumping became known as the Gruber-Durham reaction. Gruber introduced the term agglutinin. It is derived from the Latin for any substance that caused agglutination of cells. And there was a French physician, Fernand Weidel, put Gruber and Durham's discovery to practical use later in 1896, using the reaction as the basis for a test for typhoid fever. Weidel found that blood serum from typhoid carrier caused a culture of typhoid bacteria to clump, whereas serum from a typhoid-free person did not. This Weidel test was the first example of serum diagnosis, and there was Austrian physician Karl Landsteiner found another important practical application of the agglutination reaction in 1900s. These Landsteiner's agglutination tests and his discovery of ABO blood groups was the start of the science of blood transfusion and serology, which has made transfusion possible and safer. When the antibody interacts with its particular antigen, it results in agglutination, and the antibody is known as agglutinin. Antibody is known as agglutinin, and antigen is known as agglutinogen. And it occurs in equivalence zone. What is equivalence zone? Whenever you keep both the antigen and antibody in a specific medium, otherwise in a ambient medium, some of the time there is every possibility where there is a antibody excess zone or antigen excess zone. If both of them are equal, that is the zone of equivalence. In general, IgM is known as anti-agglutinin, whereas IgG is incomplete antibody causes partial agglutination. Agglutination is defined. Agglutination is defined as the formation of clumps of cells or inert particles by specific antibodies to surface antigenic components, or antigenic components absorbed or chemically coupled to red cells or inert particles. So that is direct and indirect agglutination we may go through. See here, we are observing an experiment where we have taken some test tubes containing twofold serial dilution of the serum ranging from 1:20 to 1:1280, to which equal amount of a suspension of some antigen is added, and the result of the test indicates that agglutination occurs in some of the tubes and may not be seen in some of them. Where we did not find out the agglutination, that is known as the prozone. Prozone, that is the tubes with high concentration of serum where agglutination does not occur, that represent a prozone. In the prozone, antibodies are present in X amount. Agglutination may not occur at high ratio of antibodies to antigens because every epitope on one particle may bind only to a single antibody molecule, preventing cross-linking between different particles. But here we will see agglutination of particles, where we are seeing some zone of equivalence where the antigen antibodies are found and they undergo cross-linking with each other and forming the agglutination. Here another example, same serial dilution method. The serum is same. Here we are adding 1:5, 10, 20, 40 like that up to 5120. So here the marks, the minus mark, no agglutination, and whereas you are seeing the plus mark, all these shows indicates the agglutination. Here there is a plus or minus, it is a doubtful condition, may be formed or may not be formed. That indicates that whenever the serum is more, antigen is less, no agglutination. If serum is less, antigen is more, no agglutination. That indicates that the agglutination takes place always in the zone of equivalence. Zone of equivalence. So the minimum source where actually the agglutination happens, that is known as the titer. That is 1 by 1280. So here 1 by 5, 1 by 10, this is the prozone. And we may find out zeta potential. The surface of certain particles, particulate antigens, this is also same. Here the titer is 1 by 80. Titer is 1 by 80. So remaining all no agglutination. Some of the times what happens, the surface of certain particulate antigens may possess an electrical charge. For example, the net negative charge on the surface of the red blood cells caused by the presence of sialic acid. When such charged particles are suspended in saline solution, an electrical potential called zeta potential is created. Zeta potential is created between the particles, preventing them from getting very close to each other. This introduces a difficulty in agglutinating charged particles by antigens, antibodies. So here there is agglutination reaction. Here it did not, this is antigen with the variety of epitopes antibody. No interaction in between epitope and paratope of the antigen and antibody. So no agglutination. Here antigen antibody complex has been formed, that is agglutination taken place. Here also we are seeing so this is the cross reactivity or cross linking that shows the clumping of the RBC. And the common agglutination test we will use slide agglutination. Under this, latex agglutination test for antigen detection, latex agglutination test for antibody detection, tube agglutination, Widal test, cold agglutination, wheel Felix test, Paul Bunnell test, passive agglutination, heme agglutinin test. These are the normal common agglutination tests which we will frequently use. So slide agglutination, so it is used to detect the qualitatively the antigen or antibody such as bacteria, identify and ABO blood type assays. So known antibody and unknown particular antigen on slide, that gives the agglutination. And tube agglutination, just now we discussed, that is with the serial dilution of the serum and antigen will be added, incubated and find out the agglutination. And cold agglutination, it is positive in mycoplasma, pneumonia. The patient's sera agglutinated human O group erythrocytes at 4 degrees Celsius, the agglutination being reversible at 37 degrees Celsius. So here at low temperature agglutination takes place, so that it is known as cold agglutination test. Passive agglutination, an agglutination reaction that implies particles that are coated with antigens not normally found in the cell surfaces. Particle carriers include RBC, polystyrene latex, bentonite, and the charcoal. Hemagglutination, it detects the antibody to erythrocyte antigens. If there is a sufficient concentration of antibody is present, then antibody cross-links with the antigen that forms the agglutination. If the antibodies are non-reactive, otherwise insufficient antibody is present, then there is no agglutination at all. Binding different antigens on the RBC surface detect the antibodies to antigen other than those present in the cells. So here you can observe the blood grouping. Here the red blood cell suspension. The antigen antibody, anti-A antibodies, anti-B antibodies are there. So in the A group people, A antigens are there. So here there is agglutination. Here there is no agglutination at all. In the B group people, B antigens are there. They clump with the anti-B antibody, not with the anti-A. And O group people, there are no antigens at all, no clumping either with anti-B or anti-A. And wherein the blood group AB, both antigens are present, and if you mix with the anti-A antibody and anti-B antibody, agglutination has been seen in both test tubes. And direct agglutination, the particulate antigen always combined with the respective antibody that forms the visible agglomerate or agglutination. Whereas if the antigen is soluble, we may fix it with the particles, and here there are the corresponding AB and there is a visible agglomerate. So here directly antigens are bound to the antibodies, but in the indirect the particles which are attached to the antibody, both of them, they interact with the antibody. And where we will use all these agglutination reaction, that is slide agglutination is used to identification of cultures like the Salmonella and Bordetella pertussis, typing of pneumococcus and streptococci, and blood grouping. Tube agglutination is used generally in the serological diagnosis of viral, enteric fever, brucellosis, typhus fever, that is tested by the wheel felix, blood grouping, and as well as the Coombs test. These are the normal agglutination reactions. Coming to the precipitation. Precipitation is the interaction between normal antibody and the soluble antigen. Soluble antigen. So the precipitation test may be carried out either as a quantitative or as a qualitative test. It is used for the detecting antigen and antibody. It is therefore used in application in the identification of blood and seminal stains, and also in testing of food adulteration. It can be carried out in both solutions and in gel. And precipitation test, it is defined as when an antibody interacts with soluble antigen, it will result in precipitation, and the antibody is known as precipitin. It is also occur in equivalence zone. And Marak in 1934 proposed lattice hypothesis to explain the mechanism of precipitation. According to this concept, multivalent antigen combines with a bivalent antibody in varying proportions. Precipitation results when a layer of lattice is formed consisting of a alternating antigen and antibody molecules. This is possible only at the zone of equivalence. In the zone of antigen and antibody excess, the lattice does not exist. And the common precipitation tests which are used: a ring test or interface precipitin test, slide test or VDRL test for syphilis, tube tests. Apart from that, precipitation tests in gels, immunodiffusion, single diffusion in one dimension, double diffusion in one dimension, single diffusion in two dimensions, double diffusion in two dimensions, finally immunoelectrophoresis. So here this is the precipitation ring test. It can be done by taking a small test tube containing antisera and place a layer of antigen above and the antigen antibody diffusion occurs inside the tube and forms visible precipitate in the shape of a ring here. And the precipitation curve, see here, antibodies are more, antigen is less. This side antigens are more, antibodies are less. But here in the middle we have a zone of equivalence. So where the zone of equivalence has been found, there we have the precipitation reaction that should occur. It is a graphical representation, and this is for the VDRL test which has been done in the manner of precipitation. So what is the VDRL is? The Venereal Disease Research Laboratory. The tests are slide tests for syphilis that use an antigen containing cardiolipin, lecithin and cholesterol. The antigen suspended in a buffered saline solution forms flagellates when combined with lipoidal antibodies in serum or cerebrospinal fluid from syphilis patients. So this is the slide test for the VDRL. When a drop of antigen antibody are placed on the slide and mixed by shaking, floccules will appear between the antigen and antibody. Immunodiffusion. In this method, the specific antigen antibodies are allowed to combine in a semi-solid or gel media. It is performed by using agar or agarose gel. The advantages are, it is a stable and very useful for preserving for the staining, and it can be used to detect identity, non-identity and cross reaction between antigens in a mixture. So here there is a single diffusion in one dimension. The antibody is incorporated in a agar gel in test tube, and then the antigen is layered over it. The antigen diffuses downwards through the gel, forming a line of precipitation. Precipitate will dissolve as the concentration of the antigen increases. The number of bands indicate the number of different antigens. This is the single diffusion in one dimension. And single diffusion in two dimensions is also known as the radial immunodiffusion. Radial immunodiffusion or Mancini test. Radio immune diffusion has been used for the quantitative estimation of antibodies and antigens in the serum. And double diffusion in one dimension, it is also known as the Oakley-Fulthor procedure, which is performed on the plain agar. Antibody is incorporated in gel, above which a column of plain agar is placed. The antigen is layered on top of the agar medium. The antigen and antibody move towards each other through intervening column of the plain agar. This will result in the formation of precipitate when they meet at optimum proportions. So here we can see the precipitation. And double diffusion in two dimensions. It is also known as the Agglutination procedure, where the test is performed by cutting wells in the agar gel poured on a glass slide on a petri dish. The antibody is placed in a center well, and different antigens are added to the well surrounding the center well. And after an incubation period of 12 to 48 hours in a moisture chamber, the lines precipitants are formed at the site of the combination of antigen and antibody. In this method, both antigen and antibody diffuse independently through agar gel in two dimensions, horizontally and vertically. Horizontally and vertically. There is a immunoelectrophoresis. It is a migration of molecules due to the electric charge. All the positive particles travel towards the cathode, whereas the negative particles travel towards the anode. Precipitation specificity provides a critical indicator of the identity. And precipitation reactions are extensively used in the qualitative and quantitative detection of antigen and antibody. It is a very sensitive test for detecting antigen and capable of detecting little quantity of antigen protein as 1 microgram, and used for identification of blood and seminal stains and food adulterants. Then coming to the neutralization. Neutralization is divided into two types. One is the reversible neutralization and the stable neutralization. Neutralization of virus is defined as the loss of infectivity through reaction of the virus with specific antibody. Virus and serum are mixed under appropriate condition and then inoculated into cell culture, eggs or animals. The presence of unneutralized virus may be detected by reactions such as CPE, heme adsorption, heme agglutination, and plaque formation, disease in animals. The loss of infectivity is brought about by interference by the bound AB with any one of the steps leading to the release of viral genome into the host cell. There are two types of neutralizations: reversible neutralization and irreversible stable neutralization. So, reversible neutralization, the process can be reversed by diluting the antigen antibody mixture within short time of the formation of the antigen antibody complex, that is within 30 minutes. It is thought that reversible neutralization is due to the interference with attachment of variants to the cellular receptors. Example, the attachment of HA protein of influenza viruses to sialic acid. The process requires the saturation of the surface of the virus with antibodies, whereas the stable neutralization, with the time, antigen antibody complexes usually become more stable within several hours, and the process cannot be reversed by dilution. Neither the variants or the antibodies are permanently changed in stable neutralization, for the unchanged components can be recovered. Here, the process of the neutralization, that is in between toxin and antitoxin. The neutralization tests, virus neutralization tests, neutralization of viruses by their antibodies and demonstrated in cell cultures, eggs and animals. Neutralization of bacteriophages by the plaque inhibition test. And toxin neutralization tests, bacterial exotoxins induce neutralizing antibodies, that is antitoxins, protection against diseases such as diphtheria and tetanus. In vivo tests, toxicity test, detection of toxin, C. diphtheria and Schick test. In vitro tests, anti-streptolysin O, ASO test, virus neutralization test, and nasal reaction rapid detection of Clostridium welchi. And coming to the complement fixation, let us see the complement fixation test. The complement fixation test is one of the major traditional tests for the demonstration of presence of specific antigens or antibodies. It requires a veritable zoo of reagents and numerous preparatory steps. There are almost as many versions as there have been users. The microtiter version developed at the Centers for Disease Control and Prevention includes rigorous controls and is commonly used. The complement fixation test does not depend on heme agglutinating activity of the virus, but the antibodies must fix the complement and the sera must be free of anticomplementary activity. The test has been superseded in many instances by newer tests such as enzyme immune assays. In the complement fixation test, erythrocytes are used as the target cells because complement induced leakiness of the membrane can be visualized or measured calorimetrically as an increase in free hemoglobin. In the presence of specific antibodies to an infectious agent, any complement in the system is bound, leaving no residual complement for reaction with antibodies to the erythrocytes. Thus, the presence of specific antibody is indicated by the absence of hemolysis. What are the things to be used generally? Sheep RBC suspension, hemolysin, that is a rabbit anti-sheep red cell antibody, guinea-pig complement free of antibodies to the agent of interest. So guinea-pig is the commonest source of fresh complement. And barbital buffered diluents, plastic microtiter plate, centrifuge adapter for microtiter plates, water bath for incubation of plates, and color strands for judging hemolysis. And how we will conduct the hemolysin test, otherwise the complement fixation test. Complement fixation test consists of two stages. In the first step, complement fixation stage, that is known as a known antigen and inactivated patient serum are incubated with a standardized limited amount of complement. So that is, patient serum is heated at 56 degrees Celsius for 30 minutes to inactivate endogenous complement which may disturb the test calibration. In the serum contains specific complement activating antibody. The complement will be activated or fixed by the antigen antibody complex. Antigen antibody complex. However, if there is no antibody in the patient's serum, there will be no formation of antigen antibody complex. Thus, complement will not be fixed but will remain free. Here, if you observe the two steps, here antigen is there, complement is there. Here also same, but in the serum, specific antibodies are present, but here serum without any antibody. Next step is here is a complement fixation has been done, but here there is no complement fixation. And up to this is the first stage, that is the complement fixation stage. And second step is the indicator stage, where the step detects whether the complement has been utilized in the first step or not. This is done by adding the indicator system. If the complement is fixed in the first step, owing to the presence of antibody, there will be no complement left to fix to the indicator system. The indicator system consists of sheep RBC and antibody to sheep RBC. There won't be any lysis of the RBCs. Here there is no lysis, no hemolysis. If there is no specific antibody in the patient's serum, there will be no antigen antibody complex here. Therefore, complement will be present free or unfixed in the mixture here. So, thereby the hemolysis takes place. This unfixed complement will now react with the antibody coated sheep RBCs to bring about their lysis. Then how we will interpret the result is no lysis of the sheep RBC indicates the presence of antibody in the sample serum, test serum, while the lysis of sheep RBC that indicates the absence of antibody. So this is the positive for antibody presence and this is negative for antibody presence. And coming to the Elisa. Elisa is denoted as enzyme linked immunosorbent assay. Here, an enzyme linked immunosorbent assay is a test that detects and measures the antibodies in your blood. This test can be used to determine if you have antibodies related to certain infectious conditions. Antibodies are proteins that your body produces in response to harmful substances called the antigens. Now, the Elisa is a popular format for wet lab type analytic biochemistry assays that uses a solid phase enzyme immunoassay. to detect the presence of a substance, usually an antigen, in a liquid sample or wet sample. In 1971, in 1971, Peter Pellman and Eva Engvall at Stockholm University in Sweden and Anton Schurz and Bokvan Wieman in the Netherlands independently developed the Elisa. Elisa is based on, as the name suggests, enzyme linked antibodies absorbed on the same solid surface. The most commonly employed enzyme is alkaline phosphatase, and the solid surface is that of micro Elisa plates having shallow wells. The matrix is made up of polystyrene, which has the property to bind with antigen and antibody covalently. There are different kinds of ELISA. We will perform them differently. See the direct ELISA, indirect ELISA, sandwich ELISA and competitive ELISA. Let us see the direct ELISA. What are the steps to be done in the direct ELISA? A buffered solution of the antigen. See here, a buffered solution of the antigen to be tested for is added to each well of the microtiter plate, where it is given time to adhere to the plastic through the charge interactions. Then in the second step, a solution of non-reacting proteins such as bovine serum albumin or casein is added to well in order to cover any plastic surface in the well which remain uncoated by the antigen. And the third step is the primary antibody with an attached conjugated enzyme is added, which binds specifically to the test antigen coating the well. In the fourth step, a substrate for this enzyme is added. Often this substrate changes the color upon the reaction with the enzyme. So, the higher the concentration of the primary antibody present in the serum, the stronger the color change. Often spectrometer is used to give quantitative values for the color strength. So, here we will read the color, measure the color. And coming to the indirect ELISA, where antigen is absorbed to a well in an ELISA plate. Detection is a two-step process. First, an unlabeled primary antibody binds to the specific antigen. Second, an enzyme conjugated secondary antibody that is directed against the host species of the primary antibody is applied. Then added substrate and the color will be measured. And coming to the third one, sandwich ELISA. In the sandwich ELISA used to detect simple antigen. See this one, sandwich ELISA. A surface is prepared to which a known quantity of capture antibody is bound, capture antibody. Any non-specific, second step is any non-specific binding sites on the surface are blocked. The antigen containing sample is applied to the plate and captured by the antibody. Then a specific antibody is added and binds to antigen, hence the sandwich. So above there is an antibody and below there is an antibody, in the middle antigen is struck. This primary antibody could also be in the serum of a donor to be tested for reactivity towards the antigen. Now the enzyme linked secondary antibodies are applied as detection antibodies that also bind specifically to the antibodies FC region here. And the plate is washed to remove the unbound antibodies. Then a chemical is added to be converted by the enzyme into a color or fluorescent or a electrochemical signal. The absorbance of the fluorescence or electrochemical signal of the plate well is measured to determine the presence and quantity of the antigen. So this is the third one, that is a sandwich. So below there is a antibody, above there is a antibody, and at the middle antigen is seen. And coming to the competitive ELISA. Competitive ELISA is a technique. See here, it is somewhat different when compared to the remaining one. In the first step, we will use unlabeled antibody is incubated in the presence of its antigen. Unlabeled antibody is incubated in the presence of its antigen. These bound antibody or antigen complexes are then added to an antigen coated well. The plate is washed, so unbound antibodies are removed. That is, the more antigen in the sample, the more antigen antibody complexes are formed, and so there are less unbound antibodies available to bind the antigen in the well. Hence, there is a term we will use, competition. The secondary antibody specific to the primary antibody is added. The secondary antibody is coupled with enzyme. The substrate is added and remaining enzyme elicit a chromogenic or fluorescent signal. The reaction is stopped to prevent the eventual saturation of the signal, and thereby we will measure the signal, otherwise the color of the reaction. So this is all about the ELISA, and thereby we have seen the different techniques of the antigen antibody reactions, namely agglutination, precipitation, neutralization, complement fixation, and the ELISA and different ELISA techniques. By this we will conclude the antigen antibody reactions. Thank you one and all.